Okay, we're closing out lecture 19, now talking about a process called long-term potentiation. So how do we encode and then consolidate our memories? What is actually happening in our brains to have these patterns of neuronal firing that result in recalling certain pieces of information? So Donald Hebb was a neuroscientist who famously wrote a book in the middle of the 20th century where he said neurons that fire together wire together, meaning that if you consistently send certain signals from one neuron to another, that's going to strengthen that connection. So you're kind of enhancing that pattern of neurosignaling. And so things that you use a lot, you're going to facilitate and make it easier. And things that you don't use, you're going to kind of let it decay. So the more you activate a particular pathway, the easier it becomes to activate it in the future. I like to think of this like when you make a trail in the woods. So when I was a kid, we spent a lot of time out in the woods and fields and stuff, and we would constantly be building forts, all that stuff, and then making new paths. And the first time you go through a new area, there's all these brambles and bushes and weeds and stuff, and it's really tough to get through, right? But then the more you keep walking through that area, the easier the path becomes to follow, right? What used to just take you forever and you get all scratched up now just is a breeze. So we've experienced that, right? So the fact that acetylcholine is the neurotransmitter released from the neuromuscular junction, you have heard that many, many times, and you have written that, and you have retrieved that memory, and you have answered questions about that, and you have heard me say it multiple times in this lecture already. So that neural signaling pathway should be pretty well worn in your brain right now. So that piece of information should come to you pretty quickly. Whereas perhaps at the beginning of this course, maybe you didn't even quite remember it at all and you couldn't retrieve that piece of information. So again, the more you send a signal via a particular synapse, the easier it becomes to do it again. So what is happening biologically, right? What is the equivalent of that physical difference of the path becoming clearer and clearer with repeated use? It has to do with strengthening of the synapse and making it easier to send a signal across the synapse. So we need to remember the synapse. So remember, you have your signaling or your presynaptic neuron, which is going to have its little axon terminals. Let's see, where's my pen? So they're just showing you little axon terminals of the presynaptic neuron here with a synapse on our postsynaptic neuron in this case, so some of the dendrites of it. So we have a little zoomed in view here. We're releasing the neurotransmitter, and you might remember that there's two different types of receptors that neurotransmitters can bind to. Ionotropic, which are basically just ion channels. So if the the neurotransmitter binds to the receptor, it's going to open and ions can move in or out according to their electrochemical gradient and which type of ion can move through that channel. Or metabotropic receptors, which are the ones that then activate these G protein sequence of events in the cell, which can ultimately result in ion channels opening or sometimes even other things to happen within the cell. So look at that. I'm repeating information. I'm making you fire those synapses that you first fired long ago when we learned about synapses. Oh, synapses about synapses. Oh my goodness. All right. So anyway, once the neurotransmitter binds, ion channels are going to open and the ions move. Now, depending on what types of ions are moving in what direction, you're going to get either an excitatory or an inhibitory postsynaptic potential. So again, this is review from earlier in the semester. So excitatory ones bring the membrane potential closer to threshold. So remember, the inside of the neuron is usually about -70 millivolts. Threshold is around -55. So if they are bringing you closer, I think I have an animation in here. Yeah, there we go. So let me get rid of those things. Where's my cursor? Oh, okay. So this hopefully looks familiar, maybe not, I don't know. From the beginning of the semester, I was using these little green stars to indicate like an excitatory postsynaptic potential where we've got positive ions rushing into the cell, and so it's going to depolarize it and bring us closer to threshold. And remember, if we get to threshold, then we send an action potential. So strengthening the synapse is all about making it easier for this to happen to make it easier then for this to happen. happen, right? So making it easier for the postsynaptic neuron to reach threshold so it will fire an action potential, making it easier for those signals to travel from one neuron to another. Okay, so when we talk about long-term potentiation, one of the mechanisms we're going to talk about is a mechanism for long-term potentiation. Potentiation means making it easier to do something, making it easier to send these signals that involves receptors. So we're going to focus in on a process using receptors. Particularly receptors for the neurotransmitter glutamate. So we just call them glutamate receptors. Now, these glutamate receptors that are found on neurons in the hippocampus are ionotropic receptors, so they are ion channels that will open in response to the neurotransmitter binding, and they come in two different flavors. So we have the AMPA and the NMDA. So remember, an ionotropic receptor is just a ligand-gated ion channel. The neurotransmitter, the ligand binds, they open. Okay, so let's look at what happens. These are our players. Okay, glutamate gets released into the synapse. Right, so this is their little symbol for glutamate. It's not really inside the cell. I take issue with how they constructed this. But so here's my little molecule of glutamate that is now binding to, in this case, the AMPA receptor. And in response, AMPA is going to open its channel. It happens to be a channel for sodium ions. Sodium ions are going to rush in according to their electrochemical gradient. Fine, good. We're used to seeing that kind of behavior. Now, the NMDA receptor is a little funny. The NMDA receptor, yes, glutamate will bind to it, but there's this magnesium ion sitting in there, and it is blocking the channel. So even though the ligand has bound, the neurotransmitter glutamate has bound to the receptor, so it should open, but we've got this dang magnesium ion that is blocking it. So how do we get it unblocked? Really good question. So next, as sodium rushes in, if we get enough glutamate released, right? Enough action potentials are coming down, enough excitatory postsynaptic potentials are happening. We get enough stimulation of the AMPA receptors to get enough sodium ions inside the cell, then that will kick out the magnesium. So if you reach a certain electrical threshold, so NMDA is kind of a diva. It requires not just the ligand to bind, but then you also have to get your membrane potential to a certain level to kick out that magnesium. And then if that happens, then it opens. And in this case, it's a calcium ion that rushes in. Okay, so it's like we have to reach threshold in a variety of different ways. So first AMPA in response to the glutamate binding, and then if enough glutamate binds to enough AMPA receptors, and so enough sodium ions come in, we get to the threshold for the NMDA receptor to kick out magnesium, and then calcium comes into the cell. So now that calcium has come into the cell, calcium does a lot of cool things, right? We learned previously that in the axon terminal, it binds to and activates the snare complex, which results in the exocytosis of the synaptic vesicles and releasing of neurotransmitter into the synapse. Now, in this case, calcium, which is coming into the postsynaptic cell, is going to activate some things called kinases. I'm not going to make you learn the details of those, but those kinases then cause other changes to happen in the cell. So one of those is that they phosphorylate the AMPA receptors. So if you take any other cell and molecular biology classes, genetics, you'll learn about how phosphorylation is a way sometimes we can increase or decrease the activity of different things. And what happens to the AMPA receptors when they get phosphorylated, which is they're showing you happening here, is that they become more conductive. More ion can move through. So they get kind of better at their job. It amps them up. It amps up the AMPA, right? That's a good way to remember it. And so it not only amps them up in terms of their ability to let sodium ions inside the cell, leading potentially, hopefully, if we want to send an action potential to depolarization, to reaching threshold. Yeah. But these kinases also end up increasing the number of AMPA receptors. So here's our kinases, phosphorylation to amp up the AMPA receptor, but then also we'll insert new AMPA receptors. So now not only are our AMPA receptors like souped up and better at doing their job, but now we also have more of them. So the more you use this synapse, the more those NMDA receptors are going to get triggered, the more they're going to amp up the AMPA receptors and increase the number of AMPA receptors. So the more you use that synapse, the easier it becomes to use that synapse. This is long-term potentiation. Yeah, more ion flow, more receptors. Sorry, I forgot I had those animations in there. All right, so synapses that are used more get stronger. We have more receptors and increased ion flow through them. They are amped up. Now, the reverse is also true. So if you're not using a synapse, right, then you're not doing this. And so your AMPA receptors aren't souped up. They're not particularly conductive, and you don't have all that many of them, right? So this is where we get the use it or lose it. This is why, although you formed a memory about EPSPs and IPSPs way back in, I don't know, Unit 5, Unit 3, I don't know, right? We have to revisit, right? Because it decays, memories can decay over time because then it's just like, we're not using it. Those receptors kind of go away. So this is an example of plasticity, right? So this is ways that the brain changes over time. These are structural differences happening in the cells that allow for functional differences, the ability to send signals more effectively, more quickly, more easily. Now, so remember, as we learned, we don't grow new neurons. We don't have neurogenesis. We modify synapses and make it easier for signals to be conducted in certain patterns. So, in summary, long-term potentiation, cells that fire together, wire together. If you use them a lot, it strengthens those synaptic connections and makes it easier to use those patterns of neuronal firing. So those AMPA receptors are ionotropic receptors. Glutamate binds, they open, sodium ions rush in, okay? And that's going to be an EPSP. The NMDA receptors are divas. They need not just glutamate, but they also need the membrane potential to get depolarized enough to kick out that magnesium ion. So they're kind of a two-step receptor. And then if they open, calcium comes into the cell. That calcium activates kinases, which will phosphorylate the AMPA receptors and make them super good at their job. So it's going to amp up the AMPA receptors. They can bring more ions in when they open. And it's also going to increase the number of receptors that are present on the membrane. So the end result of this is we're facilitating this synaptic connection and making it much easier to send a signal from one neuron to the next and to make that next neuron fire. So that's LTP. And that's the end of this lecture. Thanks.